Assalamualaikum warahmatullahi wabarakatuh. So this is my first video on this channel, and I make this video to meet my task about English for biology by Mr. Indra. Um, First of all, let me introduce myself. My name is Dimas Aji Bimantara, and I'm from Biology offering G 2019. So in this video, I'm gonna talk about virus. What is virus? Is it the things that make your computer run slowly? Yeah, maybe right, but me as a biology student in State University of Malang, I have another definition. What am I? So virus is a small infectious agent that replicates only inside the living cells of an organism. Infectious agent. What should I learn? First of all, you have to learn the characteristic of virus, history of discovery, structure, shapes, and replication. Characteristic. First, he's not a cell. So, is it virus is a cell? No. Why? Because virus don't have what cells have, like membrane cells, mitochondria, vacuole, lysosome, and something else. But virus have a nucleic acid, but it's just one of a kind. It's just DNA or RNA. It's have to be to bring genetic information to the next generation. Number 2, I'm living and non-living thing at the same time. What does that mean? He's a living thing. You can call virus a living thing because virus can duplicate himself. He can breed, he can multiply himself. But you can call virus non-living thing. Why? Because virus can be crystallized. Third, I'm small but dangerous. Virus is a small creature that you have to you cannot see virus through like microscope. You have to bring the electron microscope to see what virus looks like. Virus size is so much smaller than bacteria. Virus size is just 20 to 300 millimicrometer. Yeah. Number 4, I can't live without you. Who is you? You is host. What is host? So virus have to do a metabolism, right? But virus can't do that outside the living creatures, so it must be inside living creature. Next is history of discovery. This guy, Dmitry Ivanovsky, in 1892, discovered disease that caused tobacco leaf has a yellow spotting. So he discovered yellow spotting on tobacco leaves. But Martinus Willem B in 1899, that disease that Dmitry discovered is called virus. So he named the tobacco leaf virus is virus. The tobacco disease that causes yellow spotting on leaf is called virus, or TMV. TMV is tobacco mosaic virus. And then Wendell Stanley in 1935 succeeded to isolated and crystallized TMV or tobacco mosaic virus. Uh, it must be so much history of discoveries, so much smart people in here, but I only insert three of them, the first three of them, and this guy is very important for virus. Next is structure. First of all, this is a head capsule or capsid. It's made from protein. It contains nucleic acid, DNA or RNA, one of them, in here. Every unit of protein called capsomer. This is the tail, made from protein and fat. To send nucleic acid to down right over here to end plate and to inject to the host. And this is tail fibers. Tail fibers is made to attach to the host, and then end plate to inject the RNA or DNA, nucleic acid. Shapes. Viral shapes. First of all is polyhedral. Is shape of adenovirus. And then spherical is influenza shapes. Helical, tobacco mosaic virus that has been discovered by Dimitri. And this is complex that we have just learned about the structure. Complex bacteriophage. Okay, next is replication. There is two replication in virus. The first is lytic and second is lysogenic. Lytic. First of all, the virus is attached to the host and inject his DNA or RNA inside the host, and then the DNA and what is this? The core of this maybe bacteria, and then new viral components constructed. So the DNA destroy the core of bacteria and make new viral components construct. If the new viral component is construct, it makes another virus. So it can be multiply or duplicate. Host cell lyses and virus are released, and then the host cell or as we call bacteria is lysis, is broke, and then the virus can come out. That is lytic. What about lysogenic? First, it has the same step. First is virus attached to host and inject DNA, and then DNA and core right here, and then to this circle. Viral DNA integrates into host chromosome, so they becoming one. And then the host cell and viral DNA replicate and divide into two, and then when triggered the virus become active and then come to this step again. This step again, new file component constructs. This step, host cell lysis and virus are released. And yeah, some kind like that. So this is lytic. From here to here. Lysogenic, start from here, down the way up, and then boom. That's the replication of virus. And now you know the virus better. Thank you.